taking into account the biological abnormalities going on and not taking into account the specific genes that we're thinking about. And so there's a major gap there. But this is my Tuesday afternoon. This is the inner circle. And what I'm going to build is a few pieces of how that are interlocking pieces that we really have to develop scientifically, and we have to really make sure that those scientific pieces get back into the inner circle. So you've already heard very eloquently from Ingrid about the need for natural history studies and what that means, and it may start with registries. But we really need robust natural history studies that allow studies like the CLN2 Bernura study to happen, and hopefully others soon. We have to continue gene discovery. We have a number of people here who have a lot of different genes involved in their children, but several also who don't have answers yet, and that gene discovery has to continue until we solve all of these unsolved cases. We have to pursue expert variant review, and by that I mean every change in our DNA turns out not to be disease-causing, and we all have little variants from the other of us in our DNA that may or may not be disease-causing, and it really takes experts, such as many in the room, to be able to figure out what is and what isn't important, because if you have a change in a gene and there's a clinical trial for that gene, you really want to make sure that's the gene, because if it's not, maybe you're missing something else, and so that's critically important to what we do. We need to think about the functional assessment, whether it's in dishes or in animals, of, are these genes even important when we come across new genes? How do we know this is really a gene important for epilepsy? How can we tell families that this is a gene that's had an impact on your child? And then finally, as you'll hear a lot about from Steve, there really need to be robust preclinical studies, and we need to have those then lead to some of our precision treatments. And so it's very much our aspiration that all of this happens, but it does really come back to this middle circle of our patients and what we see. So at the core of all of these studies is your children, the children we see in clinics with children with epilepsy, and it's the phenotypes that we're trying to capture with natural history studies and also recapitulate in our models. So I know you all know this, but I just wanted to emphasize that we have some core definitions, and certainly there are formal classifications, but when we talk about epilepsy, we're talking about disorders with recurrent seizures, recurrent unprovoked seizures, or tendencies for recurrent unprovoked seizures, and we also think about epilepsy syndromes. And many of these for a long time have been defined by epilepsy. At some point they may be more just defined by genes or some combination thereof, but there are characteristic features of these syndromes, whether it's infantile spasms or Lennox-Gastaut or Ohtahara syndrome and others. So there can be age of onset, developmental course, EEG findings, the region of the brain involved, several things that help us sort of classify a set of symptoms into a syndrome that might point us in the direction of which gene are we looking into. And these constellation of conditions related to epilepsy are joined very much by the other features that you all know and live with, including developmental issues, behavioral issues, sometimes movement disorders, tone issues, and many non-neurological issues as well. So I mentioned this because it's not new to any of you, but it does help us to focus sometimes in terms of where we might think about honing in on our natural history data collection and also on the models that we'll develop, and I'll come back to this later. So in terms of gene discovery, I'm just going to give you a quick overview of some of the big collaborative efforts that have gone on, designed by and carried out in many ways by people in the room, and I'll just give you a couple of examples that'll lead to some specific thoughts about treatment. So many of us have been involved, thanks to the leadership of Dan and Sam and others who really got things going with something that was called the Epilepsy Phenome Genome Project, and then ultimately the Epi4K Collaborative, in which we enrolled, phenotyped, and did really exhaustive efforts across the globe to get patients together with precise epilepsy diagnoses and then to do DNA sequencing. And this started long before I was even done with my training, and I was welcomed into the community by Dan and Sam and others, and able to sort of train up in this community. And in addition to learning about the science and the science of phenotyping and also the genomic science, it also gave a young, recently graduated trainee like myself access to this amazing collaborative community. And I really want to stress that, because this is all an exercise in partnership and collaboration, and it's happening because so many people are working together. This has moved on. We had for a time something called the Epilepsy Genetics Initiative, funded partly by the NIH and CURE, and then now we have a combination of groups, both the people who were involved in Epi4K, a European group. We put those together into a joint European Epi4K group, and now we have something called Epi25 with the very lofty goal of sequencing 25,000 patients, and the collection is well on the way. And it would be a whole talk. We could hear from Sam or one of the other leaders of that another time about where that's going, but it's really, really, really exciting, because when this all started, it was an effort to get 4,000 patients enrolled, and now we're talking about 25,000 patients. And what we hope that translates into is answers for more people who are looking for answers for their their own or their children's epilepsies. So I'm just going to tell you a little bit of a story that's near and dear to me that some of you have heard a few times, but about some of the types of changes that we're looking for to illustrate a couple of features. And this is a little bit different than most of what you've all talked about in terms of the genes that are involved in your children, but it's actually looking at children who have epilepsy with a structural brain abnormality. And in these children, we have mutations that have arisen after the one cell stage. It's a somatic or post-zygotic mutation is what we call it. I'm not going to go into all of those details and can certainly talk about it in more depth at the break if anyone's interested. But the reason I bring it up is that we went from a period of seeing children who have these large brain malformations and epilepsy and really sort of thinking, well, that just sort of happened, we don't know why. And until about 6 years ago, not really having any answers, to now having answers for a good number of those patients. And we had started with very large malformations, but we've sort of walked our way down now from large malformations to very, very small malformations. And it turns out that you can have early mutations giving you a lot of your brain cells affected, down to later mutations with just a few, so bigger to smaller parts of the brain involved. And then for those of you interested in the genomics, we can have more or fewer cells affected. And what this did was, in addition to being really tremendously interesting scientifically, is that it took something that was a finding in relatively rare groups of patients. So we would see those large malformations about once a year, maybe twice a year in our academic hospital at Boston, to something that's extremely common. So focal cortical dysplasia is one of the most common causes of focal epilepsy, and we see that every week. They see it every week here at the various hospitals seeing patients with epilepsy. And so this is really an example of how really drilling down on one disorder where we thought there was a genetic cause and figuring that out has led us now to be able to solve a good number of other cases. And most of the genes that we were looking at were in this pathway called the mTOR pathway. What I didn't hear in terms of the genes represented were TSC1 and 2, and the tuberous sclerosis groups are sort of a strong entity on their own. Maybe there's some of you out there in hyperspace, but these are related disorders, and in fact we went from having 0 cases solved to about a quarter of these focal cortical dysplasias solved in a relatively short number of years, and going again from the rare to the common and really trying to extend the findings we might find in one particular study to a much broader group of patients. So I had borrowed this slide from our colleague Erin Heinzen at Columbia, and she was listing more and more genes, and we sort of finally ran out of room, and I've stopped trying to keep up with it. But as Ingrid mentioned, there are well over 100 genes for early onset epilepsies. We have over 2,000 citations in PubMed in the literature if you search for epilepsy genes. We keep discovering new genes for some of the old clinical syndromes we had, and we keep finding new phenotypes associated with age-old genes like SCN1A. And so that's all very interesting, but how do we bring this to our patients, and how do we really make this something that we can use to push the envelope for treatment? So I just wanted to mention a small publication here that was led by Lacey Smith in our group, who's here for the GETA conference from Boston. And this was really an illustration of how we really didn't think that we needed to write up what we did, but Brandy Fuhrman, who was at the NIH at the time, whom Dan had actually introduced me to through a committee, had said, this is something that you should write about, because yes, it's true the Australians are doing it, but people aren't really doing it here to the same extent. And so with homage to Ingrid and Sam for the program that they have here, and certainly with a tip of a hat to the mentors in translational science who've inspired me, like Dan. We wrote up what we do in our clinic, and it's not rocket science. It's just an example of saying that we really try to infuse with every clinical encounter this idea that we need to bring the science into it. We need to bring the clinical science into it. We need to bring the clinical genomic science to the testing world, and then we need to really push our attempts at treatment in a way that's focused on the science, as opposed to the swimming in the river approach of the old drugs that we've always had. So how are we doing that? In addition to collaborating through Epi25 now and the other large collaborative groups, we have been very lucky to have been picked as a pilot diagnosis. So epilepsy went from many of the hospital leaders saying, but that's not genetic, why are you doing genetics of epilepsy, to actually knocking on our door and saying, if you can roll 500 families in a year, we'll get them sequenced for you. It's exome sequencing, it's not genome yet, so we're a little bit behind the times scientifically there, but at least we'll get them sequenced. And we'll give you the raw data if you can analyze it, and all that will actually even pay for the patients to get clinical confirmation. So that was an opportunity we couldn't refuse. It did take quite a restructuring of our team. So Meredith Park on our team is now basically an RA who's full-time doing that, and Lacey's extra 100% of her effort is going to doing the analysis and making sure the patients who have findings that we really think are robust are getting diagnosed. And so it's a bit of a proof of principle. It's not something that's going to necessarily help us find new genes with 500 trios, but the hope is that we'll do this and be able to then contribute to the larger collaborative space in epilepsy genetics. And I mention this because a lot of what we're doing in our program really couldn't happen without the institutional support that we've had. And so when people ask, and certainly in this room, it makes a lot of sense to say, well, let's have a natural history study centered in Melbourne because that's sort of the center of the epilepsy genetics universe here. That makes a lot of sense to me because a group like this that's been spending decades building that infrastructure actually can do it, even though it might take $1 million, but they actually can because they have the knowledge and the skills, but also the infrastructure to be able to execute some of these studies. And so we're a few decades behind you, but we're trying to sort of follow in the model and do the same kind of work. And this smaller study is a proof of principle to our institution that we can actually do it with some institutional support. So with that, I'm not going to talk about the expert variant review. It's a whole topic unto itself in terms of classification and how we determine that. I really want to switch now to functional assessment and talk about how some of the models might actually be used in our labs to be able to decide, is this gene really an epilepsy gene, and to understand that gene better and hopefully move that towards figuring out novel treatments. And so again, I'll just bring back, what are we seeking in our epilepsy models? We really would like models that in some way can capture the essence of what the epileptic encephalopathies are. So we'd like to see, hopefully, some readout that represents either seizures or abnormal electrical activity. Ideally, we'd also like to be able to look at development and cognitive development and perhaps some of the other features. And in my lab, we've been trying to develop the zebrafish model. We look at very small larval zebrafish, and I'll show you some images of those, really for a number of different reasons in terms of why they're an attractive model genetically, but also because there has been a very nice illustration in terms of how this model can be a reasonable way to not only understand epilepsy, but also to screen in a reasonably high throughput fashion to find treatments. And the example comes from UCSF, from Scott Baraban's lab, where for many, many years he's spent time looking at a model of SCN1A, and found after screening many, many hundreds of fish, a compound called clemizole, which was actually previously on the market in the US as an antihistamine. It was just shelved for various reasons. Fast forward then a few years later, when clemizole, they actually couldn't make clinical grade, but they were able to find a related compound, lorcaserin, and actually deliver it through Kelly Knupp's study at University of Colorado to several patients who had some transient improvement that was meaningful. And this is sort of an illustration that this model can be useful, but also really Scott's published a number of different examples of how this can be done. So that when we were starting, there really wasn't much of a playbook. We actually went out and visited UCSF to try to find these things out, but through these publications, it's really been a nice illustration of how to go from gene discovery to an animal model, what to look for in that animal model, and then how to take that over to patients. The other thing is that because there's not only a seizure phenotype, but there are other readouts that we can have, it's been an attractive model to look at overall neurodevelopmental conditions, so we can look for seizures and seizure-like behavior, but also microcephaly, or small brain size that accompanies many of the syndromes that we're talking about, and abnormal activity in general that we're still trying to quite characterize. So I'll go through these just in terms of illustration. I just wanted to mention that it has become relatively straightforward now to do the gene editing and the genetic manipulation to model a number of the different genes. So I won't go through all that in any detail. It's sort of could be a whole hour talk, but suffice to say that the technology and the resources are available now for all of us trying to do this, that it's become very, very straightforward. One of the questions and challenges we have in the lab, though, is which genes do we want to model? And so there's a couple of different ways to address this. One of this is to say, well, what are we seeing in clinic? And how can we look at the epidemiology of the genetic epilepsies, and should we prioritize that way? And certainly we've been doing that to some extent. Lacey's been running a PCDH19 registry with Joe Sullivan and colleagues at UCSF, and we've been seeing a number of patients with PCDH19. So that was sort of one that rose to the top of the list in terms of a really deep study we wanted to do, both in terms of patient studies and developing animal models. We have a team working on that. But the other half of my lab really wanted to look more broadly and say, well, there's a lot of candidate genes. There's a lot of papers on this gene for epilepsy, and there may be one case, or there's some cellular model suggesting maybe this is a gene for epilepsy. Can we use our fish model to look at those? So we looked at the literature, and there's well over 2,000 genes somehow implicated in one way or the other related to epilepsy. We use some simple screening tools that are simple now because the Broad Institute and others who have sequenced thousands of patients without epilepsy and without childhood conditions, and all of that sequencing data those data are now available. And we can screen genes to say, well, are these genes that can handle a lot of changes in them, or are these genes that actually really can't handle a lot of changes? Are they intolerant to variation? And that's all now online. This is a major revolution in terms of, just even in the last 10 years of being able to just go online, look at a gene that you're interested in and see, is this something likely to be a disease gene or perhaps not. So that's really revolutionized our screening, and we've looked at some of the simple metrics that are available there to get our list down to something more tractable. And then because we're looking at zebrafish, we had to say, well, if we're going to do genetic manipulation in the zebrafish, it would be nice if the zebrafish gene and the human gene are fairly comparable. Otherwise, we really can't model it. And with that, we got down to a short list of about 77 genes we wanted to model. Someone asked earlier about funding and how do you fund projects like this. The NIH does not like to fund projects like this. It's too many genes, and it's a high risk sort of a thing. But it turns out that the genetic editing and the throughput and the ability of us to use our institutional zebrafish facilities and so on has made it such that we're actually about halfway through, and we've got actually a little over halfway through, and about 50 genes that we're sort of growing up and looking at. So it's tractable, but it does take some creative approaches to funding to be able to look at all these genes. But what are we looking at? How do we even approach modeling genetic epilepsy in these tiny little zebrafish? So these zebrafish that are swimming here in these wells are obviously magnified, and they're 5 days post-fertilization, and they're about 4 millimeters long each. They're swimming around here in these 7 millimeter diameter wells. And I'll show you what a seizure looks like. This is a drug-induced seizure. This is not a natural seizure in a genetic model, but this is sort of the most extreme example of what we might see. So it's swimming around in circles. It's swimming very, very quickly. And we can basically, with a tabletop device that's about yay big, look at 96 fish at a time in slightly smaller wells. And you can see these little black, hopefully gray dots that are moving around there. And those are the fish looking kind of from above. It takes a lot of student time to stare at them, and maybe you might miss one thing moving here if you're looking at 96 at a time, so we do try to do some motion detection from the machine. And we can have not only individual well data, but then we can actually look at the data in aggregate. And this is work that was started and really scaled up by my postdoctoral fellow, Jeremy Allman, but he was actually working with an old friend and colleague, Hien Nguyen, who's actually now here at La Trobe in Melbourne. So we've actually had some nice in-person meetings while I've been here to go over some of the analysis. And we're using, I never thought I would be using big data approaches to be looking for little seizures in zebrafish, but we're actually using computational algorithms and things called support vector machine classifiers to classify the behavior based on velocity and distance and various things in each of those wells to really classify seizure or non-seizure in each of the wells. So it takes something that could take hours and hours and hours and basically puts it into a 1-hour computational exercise that I can't program, but the programmers can program. So that's been quite a wonderful experience and really lovely to get to connect with them here. We don't exactly have EEGs in little larval zebrafish, but we do have some tools to be able to measure electrical activity. Here we've borrowed the multi-electrode arrays that I think you'll probably hear from Steve can be used in plates of cells, but we're basically recording as much as we can of the nervous system and getting some signal. This is work that we did in collaboration with Alex Rodenberg, who happens to be my hallmate back in Boston in the lab. And so that's one other measure of abnormal electrical activity. Another one, which we're not quite sure how to interpret, but another measure is a signal called phospho-ERK. This is a pathway that represents increased activity, and we borrowed this from our colleague Alex Shear, who's looking at a number of different genes, mostly involved in schizophrenia, but we said, there's a lot of overlap genetically, it turns out, between epilepsy and schizophrenia. What do you think about looking at, with your techniques, at some of the genes we find interesting? And so we looked, for example, at PCDH19, at these larvae that were injected, and we're finding this increased signal in these, you can see these green blobs here. So where is that? Well, it turns out that's actually the optic tectum. That's part of the visual system. So if you asked me, or if I asked Ingrid, where do you think the abnormalities might be in PCDH19? I certainly wouldn't have picked the visual system first, and we don't really know yet what this means. We're certainly going to look hard anatomically at the visual system and see if maybe, because it's expressed there, the PCDH19 abnormalities are affecting that region particularly. So it gives us a place to look in terms of looking more deeply from a neuroscience perspective, what it means about being related to seizures or not, I'm not really sure, but it's a readout that we're trying to pursue, and along with a number of different genes we can look at that. So we're really sort of trying to develop a toolkit to capture some of the abnormalities. The way we've used it in a much more simple way is just looking at head size, as I alluded to. And so we were asked by colleagues who'd found this new gene, PYCR2, which can be involved in the synthesis of something called a proline, a very basic building block in our cells. And this brain-involving synapse syndrome typically involves microcephaly or a small brain, and so we had taken animals in our lab and basically knocked down with some old technology called morpholino technology, gotten rid of this gene basically, gotten rid of the protein that it makes, and we were able to show that the brains were small. So here's just an example of the patients who have, some of the patients with these very, very small brains compared to a control individual. And what we showed in these little larval zebrafish, just by measuring the distance between the eyes, is that their heads were small. So if you look at this versus this is smaller, and we were able to calculate it over a good number of animals and show that indeed this was important. And the reason we did this is because at the time this PYCR2 gene was not known as an established neurodevelopmental gene, and so this fish data gave us some evidence that yes, it is important in the brain, it's important in brain development, and it goes along very much with the biological story that we ultimately were able to tell. Similarly, we had an experience, and this was sort of one of the many examples of serendipity that I've enjoyed in my career, but we were down in the fish room with some of our colleagues who work on kidney disease, and they said, we're looking at this kidney disease complex of genes, and our fish are swimming really strangely. They're swimming in circles. Does that neurologically abnormal to you? And we said, well, maybe, but we don't look at adult fish, and these were adult fish. Why don't we look at the larvae? And again, we asked them, what do the patients look like? And they said, well, they have kidney disease. I said, well, okay, was there anything wrong with their neurological presentation? They said, well, they have kidney disease. So we said, why don't we go and look back? And so they were in touch with their clinical colleagues, and indeed, many of these patients had very small brain size, and also some of them had epilepsy. And so we first said, let's look at the brain size. We know how to do that. That's really, really simple and straightforward, and did indeed find small brain size. And this was borne out over many animals. We did try to then look at the epilepsy, but actually the defect is so severe, at least when we produce it in our animals, that we're actually not able to grow the animals up long enough to look, even to 4 to 5 days to look for epilepsy at this point. So we may have to either move on to other variants of those genes, or just suffice to say that we know that there's a connection, but we can't really explore the epilepsy with this particular gene. So it's taught us a lot about the limitations of the model in some ways, that you can capture some features, but you can't necessarily capture everything. So some of the applications of these have come from work with other groups and looking at drug screening. We've really had an eye to, now that we've got our toolkit, we have our models, how are we going to look for drugs that might actually help a model in a fish with gene X or Y or Z. And what we have been able to do is look at brain activity mapping. This is just looking at a lot of these fish and looking at imaging where there's various signals that are increased due to increase in calcium activity and other activity that you can see pretty readily now with the imaging that's being done by many of our collaborators. So this was a collaborative exercise with a group at Mass General Hospital, and when a former resident of mine came over and said, I know you're working on zebrafish. We've done this drug screen in zebrafish, and we've identified a number of compounds that seem to help lower the abnormally high activity in some of these fish. And they all seem to be anti-seizure drugs in one category, but then there's a whole bunch of new ones. Can you help us figure out if they do anything? And so what we did, and this is basically I probably won't go through all of the details, but basically they found these abnormal activity. The blue is abnormal activity. And they found that some of these drugs, perampanel, lamotrigine, Keppra, oxcarbazepine, some of them changed the activity and some of them didn't when these fish were exposed to chemicals that made them have seizures. So you give them a chemical that makes them have seizures, and then you give all these drugs to see which of these drugs might work. So some of our usual anti-seizure drugs did work and some didn't. And then some of their new compounds actually helped. So I'm talking about 4-millimeter organisms having seizures and trying to treat those. It's not the same as children having seizures. So what are we talking about? We're talking about lots of fast swimming and making that lots of fast swimming going away. So there's a number of ways you can make that lots of fast swimming go away. One of it is just to be highly sedative and just make all movement stop. So we had to control for that, and we were able to refine this and really develop a way of being quite sure that we validated that some of the known anti-seizure medicines indeed made the abnormal movement stop. But some of the new compounds that actually came out of this screen really do seem to be potential novel anti-seizure medications. And so we've given that back to our colleagues who found them and said, it's time to run with this and see if some new drugs can actually be developed for that. So it's really helped us to demonstrate the feasibility of drug delivery in this small organism and response in a drug-induced model, which for a while we weren't sure would be valuable, but it certainly turns out to be useful and something we can do rather than in one animal at a time, in 96 at a time through these relatively high throughput assays. So we'll hopefully take this to some of our genetic models soon. I'm just going to skip this for time. And then so finally, what about the treatments? I call this individual treatments, and I've hardly talked about treatment. So I just want to go through some examples, picking up on some of the gene discoveries, both from the clinic and from our years of research, and really talk about how we can move towards precision treatment. So what are we looking for in our treatments? Of course, we'd like the seizures to go away when we think back to those core features, but when we're thinking about epileptic encephalopathy, we would like the epileptic encephalopathy to go away in the sense that we'd like to be able to reduce the abnormal electrical activity if we can. And certainly if we can modify the disease and address some of the other key features in terms of development and behavior, that's obviously the long-term goal. We also need these drugs to be tolerable, so we'd like to be more specific in our targeting of abnormalities, so perhaps we will avoid all of these unintended side effects, although we're still thinking about mouth delivery for most of our treatments. And of course, there has to be a benefit and risk balance. And we really have to think about when is it okay to just try something because it makes sense versus when do you really need a formal clinical trial. So I'll show the same slide that Dan showed, just to say that we've got a really long growing list of this, and I'm kind of running out of space, but I'm still sticking to this template because it reminds me that, when we first started this, and I listened to Ingrid give some talks at the epilepsy meetings, we knew we shouldn't give patients with Dravet syndrome oxcarbazepine, and well, I don't even think there was oxcarbazepine then, it was carbamazepine, and now lamotrigine most of the time, and we knew that phenytoin wasn't such a good idea. Now we have not only don't give those drugs, but try cannabidiol if you can get it, try fenfluramine if you can get onto a trial, a number of different options for these, and including the lorcaserin and potentially clemizole that came from the Baraban work in the fish. Many of these other genes, and I think almost all of them were mentioned, SCN2A, KCNQ2, SCN8A, PCDH19, all of you are involved in many of these genes, and KCNT1. We may not have a specific treatment that we can say we can make a formal clinical recommendation on, but we do have some things to consider, which is different than a few years ago when we either didn't have a genetic diagnosis or we just had the same old drugs that we would give anybody even without a genetic diagnosis. And so a number of these have been quite important in terms of launching projects that will hopefully lead to clinical trials, the glutamate receptor subunit genes, for example. And I'll mention a couple of examples and give you an update from last year as well. But I also want to mention, just as a prelude to Steve's work, is that we can certainly think about individual genes and the proteins they make and their individual mechanisms, but we really do have to start thinking about big picture strategies to how to address all of these, because as you can see, the gene list is going off the page and the different mechanisms are going off the page, but there may be some generalizable strategies that we can use to cover a whole swath of genes rather than necessarily one at a time. And so that's something that I think is quite important. If you've got one copy of a gene that's not making the right protein, you don't have enough of it, maybe if you can boost the amount up to normal levels, you could actually rectify some of the abnormalities that you're seeing, and you'll hear more about that from Steve in a little bit. So I mentioned that we've solved about a quarter of our focal cortical dysplasias, and all of those are in this mTOR pathway, and lo and behold, we actually have some drugs that work on the mTOR pathway from our oncology colleagues. They're called rapamycin or everolimus. They inhibit this pathway if it's in overdrive, and so a lot of my colleagues have come and said, hey, you guys have shown that this is an mTOR-related disease. Why don't we just give all of the patients, so this is about a patient a week or about a third of our patients with hard-to-control focal epilepsy, why don't we just give them these drugs? So these drugs are not entirely benign, and we don't have diagnostic certainty. We have diagnostic hunch, but there have been some attempts of putting together trials of patients with focal cortical dysplasia on these agents. I think some of the work that's coming from Melbourne, from Michael Hildebrand, who's talking about doing a biopsy just of spinal fluid, might actually help us solve some of these in the coming years. I think that'll happen, and we can be more certain. But right now, we were a little bit hesitant to just say anybody who's got one of these abnormalities on the MRI should just get one of these not so benign drugs. We didn't really have strong enough evidence to do that clinically. And then lo and behold, through a confluence of a couple of different studies really glued together by our colleague Erin Heinzen, we discovered a new gene for focal cortical dysplasia, that same kind of little abnormality, that has nothing to do with the mTOR pathway. Why didn't we see it before? We weren't looking for it before. We were looking at a small set of genes, and we were honing in and making some discoveries, but we weren't looking more broadly until recently. And what we found is changes in this gene, SLC35A2, known previously to cause epilepsy in girls and an epileptic encephalopathy, but also known in boys and girls now to cause these little lesions, those focal cortical dysplasias. And so now if you have a patient with a lesion that looks like this on the MRI, would you want to give them something that's an mTOR treatment? Not necessarily. It doesn't make any sense if it could be one of these abnormalities. That's probably more common than we originally thought. We're trying to work that out and define the phenotypic spectrum, figure out how common it really is, and really look now carefully. Maybe we can tell from the MRI, is it type 1 or type 2, but we're not sure yet. And then investigate treatment options there. So in terms of other things we can do, we certainly can do things on an N of 1 basis, meaning one patient at a time. And when we do that, sometimes we have success. So this is an example I mentioned last year. I'll just go over very, very briefly. This was a little girl whom I met, and she and her brother both had severe epilepsy. And I met them actually because they had a variant in a gene that actually has nothing to do with what's actually going on, but they had a variant in a gene. And they came and said, do you think this is involved in our epilepsy? And we said, not so much. You know, it came from one of the parents unaffected. The kids are really severe. What's going on here? And then the mom said, oh, by the way, did you know we had an exome done? And she handed me the exome report, went and got some stuff and came back, and we said, what's this gene? It's called CAD. And that day, this paper hadn't been published yet, but within a few weeks, this paper came out. It turns out this is CAD is a gene that encodes another biochemical enzyme, and it's responsible for forming a very fundamental building block called uridine. And so, well, that's interesting. Uridine has now been used in a couple of patients in Austria, and uridine is also something you could buy at the vitamin store in the US anyway. So we said, well, let's talk to our regulatory folks, but it's such a simple compound. We know it's safe. We can get it made pharmacologically. Let's see if we can give it to our one patient and measure these things. And so we did. We had diagnostic certainty. We had genetics that had just unfolded in the literature. We also had MRI progression. We knew that the cerebellum, or the balance center of the brain, was becoming smaller in her. We also had some good evidence from the blood with the difference in the blood cell size that's characteristic of this disorder. So we really didn't doubt the diagnosis at all genetically. And this is little Aubrey when she was a couple of years old, and she'd stopped being able to walk independently. And when she did walk, she was falling over and getting hurt. And we really thought the risks of not pursuing treatment were ongoing seizures, which were very, very frequent, and ongoing developmental regression. And the risks of treatment with uridine were largely theoretical, and there were some safety data already from Austria telling us it was okay. This is about a year later, sitting with the Easter bunnies, smiling, laughing, and quite feisty in clinic. We haven't gotten a repeat MRI. It's been a little bit of a production to get that done locally and justify the sedation. Her seizures are dramatically better. She was a year seizure-free when they sent me this picture of a big seizure. She's still on some other seizure medications. Her blood smear hasn't improved, so we've actually been going up on the uridine to try to address that. And I just wrote to her mom this morning saying, you know what, I should have asked you earlier, but I just wanted to give people an update on Aubrey and how she's doing. You have anything you can send me? And so here she is climbing up to go up a slide. And, you know, we're sort of every time I see a nice video of her identifying letters now with her speech therapist or something, I sort of, you know, knock everything and hope it continues, and really sort of remind myself that it's an N of 1 and we have to keep very vigilant, but also we have to look for cases like this, because even if it's only 1 in 1,000, and at this point it's probably only 1 in 1,000, it's 1 in 1,000 we can help, and the hope is that we can help more. And so this is a tale of hope, and I think it's a day of hope, and a day of being inspired by stories like this, but also humility that there's the other 999 we haven't gotten to yet. So that's that. So if it seems so simple, why is it taking so long? You know, we've got this whole system set up. Everything seems to be working, and why are we not bringing science to medicine faster? You know, what are we doing? What are we actually treating? And how do we measure success in precision medicine? And I think I want to remind us all that we have to think very carefully, as has been mentioned by the other speakers, about doing everything that we do scientifically. And we do have some cautionary tales from our own field and other fields, and really have to remind ourselves that first we should do no harm to our patients, even in our exuberance to be precise and to bring the cool science to our patients. And so with that in mind, I'll just share with you a couple of slides from my friend and colleague Tim Yu, who has been at some of the genetic meetings talking about a case of a child with Batten disease. Whoops, not sure what happened there. Did I do that? Tim was approached by a family whose child had a different CLN from the one mentioned. This is CLN7, and this is a disorder where you have to have 2 abnormalities in the gene, one from mom and one from dad. And one was identified, and there was another one presumed to be there because this child had a lot of the features consistent with Batten disease: seizures, developmental regression, and because of excellent natural history data that had been collected by a number of centers looking at Batten disease, it was reasonable to say this child had this condition clinically, even though they couldn't find the second genetic abnormality. And so what Tim's group did was they really looked very, very hard at this gene, harder than you would get from a clinical lab, because they really thought we have to saturate not just the coding portions of the DNA, the ATCG that's making protein, but we really need to look beyond that. And what they found was that in a portion of the gene that was not coding, they found a little error that was in an element called an SVA element. It's a sort of complex repeat structure that really wasn't even known about before. And so they looked, and this is something also called a transposon. These are these little elements of the DNA that weren't in the textbooks when we were learning our genetics. And what they found was that it altered the way that the protein was stitched together, making insufficient protein. So I'm sorry I can't show you the slides, but I'll just keep telling you the story for the sake of time. This is a disorder that, in addition to the epilepsy and the regression, causes some changes in storage of material in various cells, and so they were able to show that this was the case for their patient. They also then said, well, if this is a genetic defect that's not quite making the protein right, can we get in there and interrupt it and force that protein to be made? And so using strategies that you're going to hear about from Steve, they looked at a number of different possibilities for how to fix that protein, devised a clinical trial for this one patient, and were able to safely deliver through the spinal entry into her spinal fluid a medication that has helped to increase the amount of protein that she has. Now, how on earth can we go from this genetic discovery to putting something into a child's spinal fluid? It took about a year of getting animal models made, doing safety data, asking a lot of different experts in the disease and in the science and in drug delivery and in this novel drug delivery to be able to do this. And to date, it's being tolerated. The child's actually doing relatively well and is quite stable. Her seizures are stable but not gone, and we're still waiting on long-term data. So we can't really say that it's been a therapeutic success in terms of things getting better. But then we go back to the natural history, and things actually normally would have gotten worse, and we can say that she's stable, and so in that sense, it's actually quite a success. But it's also been a proof of principle that this can be done. It can be done safely. I was very, very, very nervous about the delivery and what would happen, and some of the rodent models were having some symptoms after delivery in the rodents, so we had to look for that in the patient. It's been a huge relief that there's been no harm done. But it's also involved, I would say, about 100 people over the course of a couple years to make this happen. And if you think about how scalable that is, it's not with the current resources. And so I think this is where I think we have to balance our enthusiasm and our excitement with the question of how do we balance the hope and the reality? How do we have this sort of ability to push the science and push the diagnoses and push the precision idea, but then when we figure out how to do this, how do we really then make this available, not to one patient at one institution, but to many, many patients at every corner of the world? And I think there we have to keep that hope. We have to acknowledge we have a long way to go, and we have to be humble about what we can and can't do. And so with that, I was hoping to show some pictures to thank you, but I will thank you for your attention, and I'll thank everybody who's helped me to get here, including Ingrid, who's invited me to be in Melbourne for this lovely sabbatical and an opportunity to interact with you. And I also just wanted to give a shout out. I really wish you could see my last slide because it's the pictures of you guys from last year. But I wanted to give a shout out to a local high school student, Anirudh, who's here for the second year, who's actually been educating his middle school and now high school colleagues about neuroscience and epilepsy, and really sort of give a shout out to all of you for all that you're doing to educate your doctors, to educate the young people in the community, and to keep all of us aware every moment of how important the work is that we do. And it's certainly inspiring for those of us who see patients with epilepsy. I would argue it's potentially even more inspiring to our trainees in the lab who, one encounter with a patient who's going through what you're going through, and your willingness to share that with them inspires them for years through their career. So thank you very much for that and for your attention.
Raise your hand if you have a question. Oh, yes, there's one right there. We'll get where's the microphone?
We've lost our
Amy,
microphone.
where's the microphone, people? Sorry, we're looking for somebody. Is Amy? No, Amy's not here. Oh. There we go.
Just on regards to medication, I've just gone through my daughter's medications while you were talking, because we've never been told that there's certain medications for certain parts of her seizure activity. How do I find out whether we're treating her with the right medications.
Yeah, you know, particularly with the calcium channel related genes, there's a tendency to say, well, maybe we use a calcium channel related drug, and we've certainly heard an example of that from one of our friends and colleagues in the community, Tracy Dixon Salazar, who gives a story of her own daughter who's done that. I think, you know, there's the theoretical, and then there's the balance between what theoretically might make sense, and then you've got to look at those drugs and say, well, are they safe, are they not safe? But it's still kind of at the chalkboard level. We don't actually have, for most of the genes that we know about, we don't have specific indications, you know, from the lab or from experiencing, wow, that drug shouldn't be used for that condition. So some of where these observations come from is from groups of parents who get together and say, wow, this never worked and this didn't. It's not always that simple, but sometimes it happens that way. The other thing that can happen is that if you have a group of patients and families across the world who are linked together, who have groups of physicians and researchers get together, it creates a nidus where those observations can come from. So I'm not aware with the Cas9 of specific epilepsy treatments that are necessarily going to be helpful, but I think that's a conversation that needs to be had. So for every gene I had on that list, there are 10 more where we really don't even have a place to start yet, but that doesn't mean we won't.
Jessica's on a big mixture of medications, and she still has 10 to 20 seizures a day. And our neurologist has said that he's not willing to give her any changes in her medication anymore unless something new comes on the market. So, you know, we're holding them at bay, but they're not helping.
Yeah, I mean, I think, again, we physicians have to be humble as well, and I think, and I certainly have a number of different patients in my own clinic, some of whom might be out there in hyperspace, so hello if you're there, but I don't think it's a terrible idea, and this is my own personal practice, but I don't think it's a terrible idea to have somebody go across town, or in your case, across state, if you're able to just get a fresh set of eyes. You know, we do that in our own community, whether locally in Boston or across the East Coast, every now and then, when you sort of you're at, you know, option 10 and nothing's working and you're in combination, and you sort of say, look, you know, I'm doing the best I can with the knowledge I have, but why don't we just get a fresh set of eyes and have someone take a look? That's my own personal practice. I don't think it's everybody's, but I think it's worth thinking about that, because sometimes maybe one person somewhere will have seen one patient on a combination that might have worked, and you want to give every chance possible to find that.
And do we have other questions? I'll be looking. Yes, we have a hand right here.
Hi. Sorry, my name is Johan. I've got a little girl who suffers from a WWOX deficiency. It's a recessive syndrome, very severe. She's profoundly impaired. She suffers from a nonsense mutation in one allele, and we've been looking at ways of getting some sort of read-through happening there. I was wondering if you can give us an update on your atelurin patients, and if you've seen any success, and if you've got any new insights on dealing with the NMD question.
So a couple of different things there. So in terms of atelurin, which is supposed to read through the stops, I presented a case last year, and I think, and probably the parents are out there watching, and I can connect you if you'd like, but I would say maybe partial success, but not what we were hoping for, is a summary of where that's gone, and so we're looking for other strategies. And in terms of the NMD, or nonsense-mediated decay, what that refers to is when you've got an early stop in a protein, you're just not making enough of a protein. Our observations in the past in the field have been that you just lose that protein, that it gets degraded by the cell. There have been some really interesting papers in the last couple of weeks actually published in Nature suggesting that the cell actually can kind of sense that there's not enough, and there may be actually some rescue mechanisms at play. So it may not be that you go from having one normal copy, for example, in some conditions, and a completely missing other one. You may be sort of at a mid-level, which means if you're trying to correct it the way Steve's trying to correct it, you may get more than you were necessarily thinking you would get. But I will let Steve, who's really our molecular guru here, get into that a little more with his talk.
All right, we've probably got time for one more question to try and keep us on time. Do we have another? Yes.
Me again. Just a quick question. Again, this is in reference to KCNQ2, which the natural history of KCNQ2 is a lot of the time the seizures do stop. So they're obviously given anti-epileptics to try and control the seizures in the first instance, and obviously the sodium channel blockers are the traditional ones. And then in many of the families, they try and take the medication away once they're a year seizure-free or whatever it may be. My question is, do you think that the sodium channel blockers in KCNQ2 might have a role in actually improving development as well? Like, is it just about seizure control, or is it, do the medications also affect development? Like, would it be worth staying on trilaptol or oxcarbazepine from a developmental point of view as well as just seizure control?
Yeah, so it's a tough question. It's the, as Ingrid said, that it's the important question, right? So obviously we'd like the seizures to go away, and our understanding is if you can quiet down some of the abnormal electrical activity, not just seizure related, but even in the background, that maybe you can allow the normal circuitry to develop and have some normal developmental progression. And so that's certainly the goal. So what do you do when the seizures stop? I think you have to look at a lot of different things, including, if the seizures have stopped, what's going on with the EEG? Is there still evidence of an epileptic encephalopathy? And if there is, I think we'd be inclined to say, well, it would make sense to at least remain on some agent. But again, it's still early days, and we still need to look at natural history, and we need to sort of figure out, also to try to predict who is going to stop and who isn't going to stop, and really understand those aspects of the disease. And I think the other thing we really need quite sorely is some modeling of what happens if you do and don't treat in the preclinical space, to be able to understand, does it or doesn't make a difference? If we said, well, gee, it really makes a difference even if the seizures have stopped, to stay on those agents, I don't think anybody would argue. But now, in the absence of any evidence of that, it's like, well, do you keep giving this drug for, are we doing anything? Are we causing more harm than good? And we're really in a little bit of an unknown territory that needs data. So I think we keep fighting for more data to inform us, because you still have to do something. We were talking at the break, even if you don't have a gene to define a condition, you still have to do something. So you make an educated guess, but we're at the point now where there's so much science in our field that we would like to be able to do more than just an educated guess. So I think in 5 years, I hope we'll have a better answer for that.
We've got one more question for you, and then we'll let you sit down and go into our next speaker. This is from somebody who's watching us online. Thank you very much. This is from Kim in Adelaide, and she says, children labeled as idiopathic, should they have repeat genetic testing as things evolve?
So that term, our classification gurus I will tell you, is no longer used among many of us, because we've favored conceptually that idiopathic is really presumed genetic, and what was previously formal, and I mean that not to say that you've got the wrong term, but just that we as physicians, when we put these terms on them, didn't necessarily know quite what to call them in the early days. So what we used to be called idiopathic generalized epilepsy, for example, then moved on to being called genetic generalized epilepsy, because of strong evidence, much from here in Melbourne, that there's a genetic basis. It hasn't been quite so easy to figure out the exact genetic basis for many of those, but I think the strong presumption is, yes, there's going to be something genetic. So should you keep trying? You should keep trying, but this is where the research that's being done here and many other places is so important, because there aren't a lot of clinical tests yet available for most of these previously labeled idiopathic disorders. And so the best way really to move that is to be involved in research studies that will then collectively, over many thousands of patients, lead to that knowledge.
All right, thank you very much, Anne Poduri. And we have, here you get your chocolates, and you get also an Australian book as a visiting American.
Thank you all.